Thank you, Ben. And thanks to all who are attending, taking the time to attend today. It's a great pleasure to have this opportunity to talk about what we've been developing over the years and are currently bringing to market. As a publicly traded company, Angle PLC, I do need to share this disclaimer slide. I will be presenting both published data as well as some unpublished results. None of this information is presented in the context of direct use for clinical or diagnostic purposes. And I would just ask that none of the unpublished results be reproduced in any way, but please feel free to ask questions. As Ben mentioned, this particular format doesn't allow for questions during the presentation, but if you can type them in, we'll do our best to answer them within the time frame allowed. So once again, thanks for joining. This is most likely a little bit like preaching to the choir in terms of the audience. Certainly the value of liquid biopsy is clear. And although it's been a popular term lately, certainly liquid biopsy in one form or another has been around for a long time. And importantly, as therapeutics have advanced in their specificity, it becomes even more important to be able to repeatedly monitor a patient and in the field of oncology and understand the changes that their cancer is undergoing, either in progression or response to treatment. So having access to a relatively straightforward process that is non-invasive to the patient will really facilitate the effectiveness of those drugs. But one of the key questions is how does this turn into routine practice in the clinic? If we look at what it might take to encourage broad adoption of liquid biopsy, clearly we're still on the learning curve. The use of sequencing is generating new information literally every day and with every sample in terms of the changes associated with progression of cancer. But there is potentially a need, particularly in routine monitoring, to be able to run simpler tests that can deliver actionable information that can be associated with treatment of that patient. And it's important that this be delivered close to the patient in a rapid turnaround time so that not only the treatment can be effective, but the anxiety of the patient at a particularly critical time can be alleviated by having less waiting. Obviously, non-invasive and affordable are very two key attributes necessary to roll this out. But if we look at it also from the laboratory perspective, the lower the complexity of the test, the simpler the laboratory that can be run in, the broader the adoption that will take place. Ideally, no lab likes another box on the bench, so the more types of assays that can be included in these platforms, the more practical it is for adoption. And it should ideally be able to adapt to the rapidly changing market that we're facing and the wealth of information in terms of treatment of cancer and new biomarkers as they come out. If we look at what we're going to spend a little time on today, effectively we're going to examine parts of this workflow from the original blood sample through to the isolation of CTCs and other rare types of cells, and the subsequent analysis of those, either through traditional techniques such as imaging or some newer approaches to examining drug therapies and their impact on captured cells. But the main focus today will be on molecular analysis of those CTCs and other rare cells that are harvested from the blood sample by the Parsortix instrument. But it's important not to forget that once you retain the cells, there's a lot of very interesting information effectively left in the sample in the form of ctDNA, particularly useful in examining actional mutations, exosomes from a signaling perspective, and my personal favorite, given my experience, soluble proteins, the traditional tumor markers and other biomarkers of that nature that are in the blood. An important consideration at this point, depending upon what targets that one is planning and looking at, is the pre-analytical treatment of that blood sample. Depending upon whether the goal is imaging, one may want to protect the cells in one form or another, either prior to harvesting or in the harvest itself. One may want to protect against degradation of circulating tumor DNA or stabilize the RNA in the sample, both within the cells of interest and in the broad cell population that's present. So it's important to look at and consider that as the sample is collected. And it may make sense to aliquot part of the sample to different types of analysis from the outset prior to isolating the cells. Typically, isolation of CTCs is going to take the largest volume of sample because there are so few present in that blood. But as you'll see, most of the other analytes can use just a tiny fraction of the sample for analysis. So what we will focus on today is the workflow that we at Angle have put together for sample-to-answer multiplex testing. And this really consists of those three components that we just talked about. The Parsortix instrument uses a microfluidic device to separate and harvest rare cells. The HyCEAD chemistry is used to enrich and amplify the particular gene targets of interest in a multiplex format. And then the Flow-Thru chip is used to detect those multiplexed amplified targets in a rapid and highly sensitive fashion. And this, as you'll see, can be carried out in a variety of techniques, both automated and manual. So let's start first with focusing on the capture of these CTCs. The Parsortix system uses a epitope agnostic approach for capture of CTCs. In particular, cells are selected based on a combination of their size and deformability. As you'll see in the animation to follow shortly, basically blood directly from the collection vacutainer, and all of the data that you will see here today comes from a standard EDTA type blood collection. That tube is mounted on the instrument, and the blood is processed, is flowed through a cassette, and cells that are present are directed towards what's called a terminal gap, shown in the center section here. And small cells such as red blood cells, will rapidly pass through. Larger cells, such as white blood cells, at the appropriate applied pressure and with the appropriate gap, will squeeze their ways through. While cells that are less deformable or larger, which is typically characteristic of CTCs, will be trapped in advance of that. Flow can then be reversed, and those cells captured in either the pooled sample with rare cells, as well as any residual WBCs, can be analyzed directly, or they can be further processed, individually picked for single cell analysis. So this is a little better idea of what is happening within the cassette. basically as blood flows through a channel from the inlet, it is allowed to flow up and over and out the terminal gap of a series of very small steps. This gap size is selected for different applications and ranges from 4.5 micron up to 10 micron. In most applications for CTCs, 6.5 micron gap is used and is adequate for collection. Pressure and gap size are two variables that one can use to bias selection towards larger or smaller cells. And basically, in this animation, you'll be able to see exactly how the separation takes place. The cassette that is shown, to give you a perspective of size, is identical to a microscope slide. And basically, the video will be effectively looking down the channel of the terminal gap. So the roof of the video, if you will, is that terminal gap. You can see blood flowing into the cartridge and up and over the steps and out the terminal gap to waste. This can be followed by a wash buffer to remove residual materials, and then the flow reversed and the cells captured in a harvest vessel. Alternatively, these cells can be stained directly in the cassette, which again is sized to fit in a standard microscope slide apparatus. If we look at this in sort of more detail, an actual video of blood flowing through the cassette, here the wide lighter portion is that terminal gap. So we're now looking down on top of the cassette. You can see in particular the red blood cells climbing the steps and exiting into the waste. And basically, as the blood processes through, any particular cells that are larger than that gap, as shown here, will begin to accumulate at that stage, and then can be either imaged directly in the cell by addition of staining agents or flushed automatically backwards into a collection vessel for further analysis. So that's effectively how the platform works and captures those cells. The time of blood processing varies depending upon, obviously, the volume of the sample, the gap selected, and the pressure applied. But typically is in the range of half an hour to an hour and 30 minutes for anywhere from a 5 to 10 mil blood sample. The harvest is collected typically in 120 microliters of buffer, and effectively you have a very high enrichment of these targeted cells that can then go on to be analyzed, again as a pool, or if single cell analysis is desirable, it provides a much smaller volume and enriched fraction for the various sorting techniques that can be used to pick these cells and isolate them. So if we look at some of the data that reflects these types of experiments, and I would recommend that on the Angle website, there's a good selection of literature that looks at different cell types, various publications of how the process has been applied. But one of the key messages is shown on the left-hand side, in that you get a pretty good reduction in white blood cells, along with the enrichment of the targeted rare cells. And that would range anywhere from a couple of thousand WBCs up to maybe 5,000 or 10,000 maximum, out of a 10 mil blood sample, along with any captured cells. And that will vary from patient to patient, based on the characteristics of the blood. But what hopefully you'll see in the rest of this presentation is that doesn't necessarily have an impact on the analysis of those rare cells. One key factor is that the efficiency of that collection is it's definitely different for different cell types. That's effectively a characteristic of this recovery. So depending upon size and deformability, one will be more efficient at collecting one cell type versus another, as shown in the bottom right-hand slide. However, it is epitope independent, so what has been well established is that an additional population of cells, CTCs, are collected in addition to traditional EpCAM positive cells. At the top, this is an example of harvesting of SKBR3 cells spiked into healthy normal volunteer blood, and showing recoveries in the high 80% range. But again, this will vary depending upon the type of cells and the purity that is desired and the parameters applied to the system. There is a distinct advantage clinically often in being able to collect cells beyond epithelial cells. And that's shown here in this excerpt from a publication from Bart's Cancer Institute here in London. In this particular study, they were looking at prostate cancer patients, isolated CTCs using the Parsortix system, and then stained them for nucleus, CD45, cytokeratin, and vimentin. So the sort of typical markers to differentiate circulating tumor cells. And you can see in the top row that they indeed did identify and isolate epithelial type cells, but also were able to capture mesenchymal cells and those that are in transition. And as it turns out, that ratio was particularly helpful in understanding the progression of survival. Another interesting observation in this particular studies is that they isolated some very large cells, which were subsequently identified to be megakaryocytes, shown here in the bottom. And the presence of these cells correlated with a positive outcome for these prostate cancer patients. And the combination of the ratio of these various types of cells, along with the numbers of megakaryocytes that were present, could be used to separate and classify patients into good prognosis in terms of survival over time, post-analysis of these samples, and those with a poor prognosis. So very encouraging work, which is ongoing in larger studies, but showing the value of capturing a broad range of cell types. Another researcher using the Parsortix system, in this case at USC Breast Cancer Program in California, did an interesting study looking at patients with metastatic breast cancer, and captured both CTCs at the same time that metastatic sites were being biopsied for tissue, and then performed RNA-seq directly on those harvests from the Parsortix instrument, and compared the genes that were present and their expression levels to the matched biopsies from the metastatic sites in each of those patients. And a couple of things jump out over here in the plot on the right. May be difficult to see because of the size, but effectively all of the peripheral blood RNA clusters together, whereas the metastatic sites in the CTCs were clustered as a group with some degree of overlap. And indeed, when you look at the individual pathways, there was no significant difference between the CTCs and the metastatic biopsy analysis, which is very encouraging in that it might provide a window into pathway changes and potentially the use of this to follow and modify therapies without the invasive process, particularly of individuals with metastatic disease, to have sequential biopsies. And in this particular work, Dr. Lang identified over 60 pathways that had some relevance to new or existing cancer drugs. And again, this work is ongoing. So, large number of ways that these harvests can be analyzed, again, either on a pooled basis, with staining, with culture, or processed with additional cell picking technologies to be able to look at individual cells. Our focus has been towards developing platforms that could eventually be used on a distributed testing basis, as I mentioned in the outset. So the Parsortix instrument is relatively straightforward to use. The vacutainer of tube of blood is attached directly and the cells harvested. So if we can also develop molecular analysis that can be done at a local level in the local hospital lab, and long-term potentially in group practices, then that may offer some advantages to being able to deliver this information to the patient. So what we developed was something that we call HyCEAD, stands for Hybrid Capture, Enrichment, Amplification, and Detection. And in reality, this was developed to deal with a common problem in nucleic acid analysis, particularly multiplex analysis. There are lots of good multiplex detections platforms out there. We happen to have one, what we think is particularly in the flow-through array, that's particularly sensitive and rapid. The challenge has always been to do multiplex amplification and not have interference between targets, or not have to do a lot of optimization to get all the targets to work together. So really, this was a method that was developed specifically to move the, if you will, the multiplexing out of the amplification stage into the sample pretreatment stage. And effectively, what this does is we start out with the sample directly. So for instance, a Parsortix harvest, there is no pre-extraction of nucleic acids. Instead, in the lysis buffer directly are a number of gene-specific primers that target the regions of interest that are to be measured. These are allowed to bind to their targets of interest, and then these complexes are pulled down onto a solid support. And depending on how these are designed, we can target either intact RNA or fragmented RNA or DNA, depending on the design, but the process remains the same. Once they're on the solid support, that gives us the opportunity to wash away to potential interference. It also gives us the opportunity to wash away the high concentration of primers that can be used to drive that binding very quickly. So then we can introduce a series of enzymatic steps under very favorable conditions to be able to effectively copy those sequences of interest, bracket either side of the sequence of interest with universal primers, introducing those in the copy phase and extension phases. And it is designed so that at the end of this process, the amplicon, one representing each target, with a copy of the sequence of interest bracketed on either end with a forward and reverse universal primer, is released into solution. All of this takes place on a single solid support so that we can automate this very easily. So it's a sequence of solutions that are passed across a solid support. This can be automated manually with magnetic beads, on a robot with magnetic beads, or in a cartridge, as you'll see later, on a different type of solid support. Once those amplicons are released, no matter how many targets to the PCR, they effectively look like the same target. They have the same universal priming sequences. The targets are selected to have very similar melting points, GC contents, etc., so that they can all be amplified together in a single PCR condition, or similarly with particular isothermal amplification strategies. And what this means is that we can determine or design assays with different targets just by changing the primers that are added to the mix, the probes that are added to the mix. We do not have to re-optimize the amplification or the assay conditions independent of the content in the assay. Once these are amplified, and the data that you'll see in the next few slides, typically we run about 30 cycles of PCR, and then these are hybridized directly on the flow-through chip in order to quantitate the amount of material present. So how does this assay perform? So on the left-hand side, if we look at reproducibility studies, in this case with 27 cycles of PCR, here we have taken cell line RNA, in this case 50 picograms of CaOV3, and spiked that into a background of 50 nanograms of WBC RNA to mimic more closely what one might find in a harvested sample. And you can see for this 25-plex panel, the reproducibility is very good across the range of targets, and typically we're seeing under 20% CV for the process from lysis through to the final result. In terms of sensitivity, we've developed a series of synthetic mRNA controls that we can quantitate very accurately and spike those into samples in order to both control the assay and make sure that all of the steps have behaved properly, but also to, if you will, characterize the assay in terms of its sensitivity and susceptibility to changes in the assay. So in this particular case, we were spiking in very low levels, a few molecules of targets, and 4 of them are shown here. The 4 blocks at each data point represent 4 replicates. And I will explain that the scale on the bottom, the reason, which is actually the number of input molecules, the reason those aren't round numbers is because as you dilute these standards down from the original estimated concentration based on absorbance of a concentrated solution, obviously the accuracy isn't good enough at that level to be this precise. So instead, those spiking solutions were subjected to digital droplet PCR to confirm the exact number of molecules that were in each spike. So for an example, the top left-hand panel is green fluorescent protein, and you can see there that with 0 spikes, we got 0 signal, and for 8.5, 17.1, and 34.1 molecules, or the equivalent amount of RNA to that, based on ddPCR, we got signals in all 4 replicates. And some targets are more sensitive than others, some less sensitive, but generally certainly for 10 molecules or more, we always see a signal for all replicates as an input, and generally 5 molecules or less below that, we start to see stochastic behavior where we will get some hits and the rest being dropouts. So we're very encouraged by the sensitivity, given that this is in the presence of either spiked into blood or in the presence of high levels of WBC RNA. Similarly, if we look at this kind of experiment step now spiking in cells, and in this particular case, the cells are actually counted under the microscope before going into the blood. So they're, again, CaOV3 spiked into healthy normal blood. What you're looking at here is the number of cells spiked into the original blood sample based on the actual count going in. the non-template controls, the 0 spikes, and then a range of samples from 1 to 6 cells. And these are all individual genes across here. You can see on the right-hand side that there are some genes that are expressed at low levels in the WBCs, so in the no spike controls. But we're not, in this case, getting any signals for the more cancer-specific genes, the more cell line-specific genes that are shown on the left, and we get an increase with the number of cells that are spiked in. Apologize how complicated this slide on the left is, but very quickly, what this is showing is that with the right selection of genes, we can actually get specificity between cell lines within these samples as well. So each of these numbers down here represents a different gene in the panel, and if all 3 cell lines spiked in, so these are individual blood samples with cell lines spiked in, cell line RNA spiked in. Sorry, these are not blood samples. These have now 500 nanograms of WBC, depending on your math, equivalent to 20,000 or more WBCs with 25 picograms of either RMG1, CaOV3, or SKOV3 RNA spiked in. So if they all record a signal, you will see a cluster of 3. The takeaway is with appropriately selected genes, there are definitely targets that can discriminate between the various cell lines, some that register to all of them. And on the right-hand side is really the proof of the pudding, if you will. This is a 47 gene panel presented in a radar plot fashion. So effectively, there's each of the genes from 1 to 47 around the outside, and the intensity of those signals increases from the middle outwards. The white line is the gene signals, the intensity signals for the unspiked sample, and the blue pattern represents the pattern of gene expression for the spike sample. And the actual fraction, because not all of the sample is analyzed here, that generated this data was equivalent to the mRNA from about 1.5 cells in the original blood sample. So good specificity. It's clear that we can detect the presence of a very small number of CTCs in a background of WBCs in a Parsortix harvest. And if we look at reproducibility of the entire workflow, so this includes processing the blood on the Parsortix instrument. So here we have 3 different donors, each who gave 4 tubes of blood, and processed, spiked with the same number of cells, processed all the way through Parsortix, HyCEAD, analyzed on ZipWell. And each of these plots represents the gene expression pattern from the 4 replicates, and you can see the concordance between those 4. And the median CV in this particular experiment was 28% for 25 genes through the entire process. So that's how we prepare the sample. How we analyze the sample is using the Flow-Thru chip technology. And basically what this represents is we have a small silicon substrate, which in the version shown here is mounted on the end of a pipette tip, if you will. And that substrate is completely porous, so it has a series of very uniform holes that go right through its 350 micron thickness. Those pores start out at 10 microns on the face and then taper down from there to the backside. And they're on a very tight pitch, 12 microns. So if you hold this up to the light, you can actually see through the chip because of the open area. That chip is printed like a normal array, so spotted with, in the case of nucleic acids, an oligonucleotide capture oligo that covalently binds to the surface and represents, each spot represents the complementary sequence to a target of interest. On a chip this size, we can print a little over 500 spots with replicates and controls, 125 to 150 targets. The difference between this and a standard array is the fact that each of these spots, which is about 100 microns, similar to what you would get, for instance, printing on a slide, actually covers the walls of about 100 of these channels all the way through the chip. So we get this massive increase in surface area. And as we pass the sample back and forth through the array, it gives an opportunity to get much more efficient binding of targets from solution to the immobilized capture probes on the surface. And that's both due to surface area, plus the fact that these pores being only 10 microns wide, the most anything has to diffuse to get to a binding partner is 5 microns. We like to call it speed dating for molecules. So we get a very efficient capture as it passes through the chip and effectively overcomes the diffusion limitations that are typical of slide-based arrays. You'll see here in the last couple of slides that this array, actually the substrate plays more of a role than just an array. We actually use it to control the fluid flow in our devices. It acts somewhat as an optical guide to enhance the signal that we get, and can also be used as a solid support to perform things like the HyCEAD sample prep that we just went through. So very quickly, this is how the ZipWell platform shown on the right-hand side works. So the instrument picks up a 1 to 8 tip chips, as we call them, that are printed with the capture oligos, dips them into a sample or a wash or a block, whatever the assay steps that are required, and then passes, applies a slight pressure and vacuum to pull that sample back and forth through the array. And at some point in our amplification, we incorporate a biotin into our targets. And towards the end of this process, the captured targets are reacted with streptavidin HRP enzyme, which binds stoichiometrically to the biotins on those targets. The very last well contains a luminol substrate with a camera underneath, and when the chip is processed in that luminol, the HRP processes the substrate, generates chemiluminescence. We capture that light in a series of exposures, and it's proportional to the amount of target captured. You will have noticed that that flow is quite uniform going back and forth. This is not a volumetric process. There isn't a series of syringes on the other end of this. I mentioned the pores are actually used to control the flow, and they're designed such that at the pressure drop we apply, the pressure differential, only fluid will pass through the chip, not air. So it'll pull up whatever liquid is available and stop until we reverse that pressure. And we use that in higher throughput versions and eventually in a cartridge version of this platform to greatly simplify how the instrument and how we can control reactions. So this is an instrument that is currently under final development. We call it the ZipWell platform. And from a high level, basically, instead of moving the array to a solution sequentially through each step of an assay, we have adapted this to fit on a standard liquid handler type device, and we can deliver the solutions one after another to the array. And incorporated in this is the ability to aspirate that solution off automatically. This makes for faster and more efficient platform and fits a standard 96 well plate format, 48 of these devices to the footprint of a plate, and delivers improved sensitivity due to some other enhancements on the way through. So that's what's used to detect all of the nucleic acid products that we produce. This combination of HyCEAD and ZipWell detection can really be adapted to a very wide range, and working with partners, we've demonstrated a wide variety of applications. So with the appropriate probe sets and chips, we can look at mutations, again, in a targeted panel basis. We can look at fragmented RNA in FFB tissue as an example. We've done work in sepsis for the rapid detection of sort of an immune profile based on changes in expression in white blood cell RNA. viral detection in blood, in particular HIV, given the high sensitivity, and working directly from a crude sample. There's some advantages there. And one really good application outside of oncology is in infectious disease. Given the ability to see such a small number of targets directly, the no need for extraction of nucleic acid first, and the fact that we don't need to depend upon multiplying a commonly preserved gene like 16S for multiplexing, that allows us to look at a wide range of targets together, and also allows us to look at things like a wide range of resistance genes within the same sample. As I mentioned earlier on, I'm a bit of a protein person, so I had to sneak this in. The nice thing is that without any sample prep, without using the HyCEAD technology, we can do protein analysis directly on the ZipWell or the ZipWell platform. That binding efficiency leads to very high sensitivities, sub-picogram per mil, low femtogram in particular cases with good antibodies. Particularly well adapted to things that use a common detector, such as allergy, anti-IgE, vaccine status with anti-IgG or IgM, etc. And we've demonstrated wide compatibility even with things like urine directly in the well of the plate through the tip chip, and the sensitivity allows us to use very small samples. We're fortunate that last year we published this study on traumatic brain injury related injuries, in particular intracranial injury in infants. And over the last year I've had the support of the Coulter Foundation to expand that trial into older children up to the age of 4 to help screen for the presence of brain injuries and validate whether or not imaging would be of interest. So just the final slide, where we are from a commercial perspective. The Parsortix platform is currently in a multi-center FDA trial to submit for potential clearance as a device to capture CTCs initially from metastatic breast cancer patients, with secondary endpoints for the use of multiple downstream analysis techniques, from imaging to FISH to NGS. The HyCEAD ZipWell platform, we completed our first large clinical study in ovarian cancer, specifically looking at gene expression profiles to potentially triage patients with pelvic mass to the appropriate surgeon. And a subsequent trial to follow up on those successful results is about to get underway currently. We have both manual and automated platforms available at this point, focused predominantly on both collaborative work with interested parties who are looking to develop clinical applications and clinical data that is sort of in our sweet spot of targeted panels, so gene expression panels, mutation panels, etc., and also working with partners outside of our core focus in liquid biopsy and oncology in areas such as fetal cell isolation, infectious disease, etc. And although this was a quick pass through, you can hopefully see the promise, and one of the purposes of all of this work is to eventually combine all of this into a single cartridge that takes advantage of all of these technologies that are effectively driven by a small pressure differential to put it into a common instrument and really move this out to the point of deployment for testing. So thank you very much. I will stop there and take questions as available. Thank you.
Thank you, Paul, for your fantastic insights. As mentioned, we do have some time for some questions. So if you would like to submit a question, please do so in the question box. Paul, here is the first one for you. Where do you see this technology positioned relative to sequencing?
Thanks, Ben. Yeah, that's a great question that we get a lot, and I see it as complementary from a couple of perspectives. Certainly, sequencing has a lot of advantages, and whether that's genome-wide in terms of discovery or targeted sequencing of particular genes of interest. But it provides a wide range of information that might be difficult to execute or to put to use without interpretation. We are, and that doesn't diminish its value, it just puts it into a different category of deployment, if you will. We are really focused on targeted panels that are associated with sort of known actionable mutations, or in the case of gene expression, panels that have been developed to answer a specific clinical question. And probably the best known of these are things like breast cancer prognosis. And in that particular case, the answer you get depends upon measuring some very specific genes and their relative expression in order to calculate, in this case, the risk of recurrence or the value of a particular therapy. And we see that a good opportunity within that range. We can adapt this technology to quickly deploy a variety of panels as they come out. In some ways, I see the, if you will, the number of actionable panels is to a degree limited by the rate of drug development, so that there will be, in terms of panels associated with particular new drugs. So there will be a need for targeted panels for a long time to come as all of those drugs work their way through the pathways. In a general sense, there's potentially advantages versus sequencing in terms of being able to deploy this in a less sophisticated lab, so not necessarily needing a centralized lab with an extensive IT infrastructure, and significant cost advantages to being able to run the assays in this format.
Thanks, Paul. A second question here for you. In the context of the HyCEAD process, what did you mean by standardized process facilitates the introduction of new targets?
Perfect. Well, somebody was listening. That's good. That's quite a catchphrase, but really, the explanation for that is the fact that we have developed a process to introduce effectively universal primers onto either side of a sequence region that is chosen to behave similarly in a PCR reaction to other chosen sequences. That means that we can substitute in and out genes by just changing reagents. So we do not have to change the workflow. We don't have to re-optimize the PCR, none of the conditions for analysis. We effectively just change the solution mixture in the set of tubes that comes in the kit, and we provide a chip with the appropriate capture probes printed on it for those targets. And we've demonstrated that in real life in terms of mixing and matching. We've developed quite a number of gene sets relative to reagents, and demonstrated the ability to sort of mix and match those at will to address particular questions.
A third one here. You mentioned different applications towards the end of your presentation. Using this technology in other biofluids, are there any nuances with performing and analyzing the test?
So a great question, and obviously there's always nuances. We actually started out developing this in the infectious disease area in terms of the first project that we worked on. It happened to be HIV, and we're relatively surprised at the ease with which that translated to captured cells and harvests. Not to say there hasn't been a lot of work by the scientists on the way along, but effectively, I think the key parameters are with different sample types is we are doing a hybrid capture in solution. So the sample has to be compatible with an effective lysis buffer. There can be, so for instance, a tissue would have to be digested to make the material accessible to solution for the capture to take place. But it's pretty tolerant of debris, if you will, in the process. So that's really the key item. We have shown, we've looked at a wide range of sample volumes, a wide range of sort of conditions of those samples relative to ionic strength, concentrations, etc., and basically have developed a standardized process that's pretty tolerant to the sort of range of physiological conditions that one might see in these various sample types.
Another question here. What is your commercial approach to the market?
Thank you. Yeah. Sort of as I mentioned a little bit towards the end, this is really about focusing our activities on targeted panels. So how that translates commercially is clearly if we're targeted panels, we don't have everything every researcher wants or might desire. So we need to either for initial studies, proof of principle, if you will, use genes that might be relevant from lists of probes we've developed, or work with them to understand probes and targets of interest to put together a custom chip. And internally, obviously, we are working our way through to be able to provide a true sample-to-answer solution. So we are developing some clinical content directly ourselves, but with other applications, we work with and look for commercialization partners who would have access to the appropriate channels for those markets. We are internally, if you will, and direct to market focused on liquid biopsy and oncology and things like fetal infectious disease, allergy, etc. Look to work with partners to develop those applications and commercialize those. We do currently offer contract services that for sample analysis for either initial work or sort of development research projects for both academic, clinical, and pharma customers.
We've got time to squeeze one quick question here. Last question, Paul. When you isolate a cell after Parsortix and Deparay, how can you make sure that it's in fact a CTC?
That must be a trick question. I don't think there's a perfect answer to that, but there's a wide range of approaches, and I'm not exactly the one. I'm certainly not the one to speak to all of them, but this can be, is typically confirmed in a variety of ways, mostly through staining and identification through either surface cell markers or interior markers through a staining process. But as I also alluded to very briefly in that rather complicated slide, it is potentially possible to characterize through the appropriate selection of genes what cell type it is. And we've done some work looking at sort of broad range of cell lines to try and identify targets that can differentiate closely related cell lines in things like ovarian cancer where the type of cell has that is present is indicative of potentially very aggressive versus less aggressive disease. So certainly there is enough material in a single cell to be able to analyze with HyCEAD, and in fact, the background is a little less without the WBCs there, so one might even be able to see genes that we can't see, even with the sensitivity we have now. But that is one approach to characterizing. The traditional approach would be an imaging step at the point of isolation prior to, for instance, sequencing or HyCEAD analysis on a single cell basis.
Thanks, Paul. That is all we have time for. Once again, I'd just like to say a big thank you to our presenter, Paul Smith. We hope you've enjoyed the session. If you do have any follow-up questions or comments, please do email me at ben.carrington@hansonwade.com. Just as a reminder, a link of the recording of the presentation will be emailed to you all in the coming days, so please do look out for that. Other than that, it's just left to say thank you very much for your time. I hope you have a great rest of your days and have a happy holidays as well. Thank you.